23rd को हमारी लास्ट क्लास हुई थी। क्या किया था हम लोगों ने? ट्रांसपोर्टिंग प्लांट रिवाइज किया था।
यस सर।
यस सर।
आज हम लोग क्या करने वाले हैं, क्या रिवीजन करने वाले हैं बताओ।
सर कैन वी रिवाइज सेल साइकिल?
हाँ जी बेटा,
सेल साइकिल
रिवाइज कर लें। चलो सेल साइकिल करा देते हैं। ठीक है, दोनों लोगों को सेल साइकिल ही करना है ना। चलो सेल साइकिल कराता हूँ पहले। देखो एनसीईआरटी अपने साथ में रखो। सो कीप योर एनसीईआरटी विद योरसेल्फ। सो let's start with the cell cycle and cell division. Look, before going into cell cycle and cell division, try to understand this quickly. So here, what is actually cell cycle? Cell cycle is complete process of division of a cell, right? So what happened? Suppose that formation of a cell takes place, a new cell, right? So look, here, this is the new cell. So formation of a new cell takes place, and when a new cell is formed, after the formation, it will go for a maturity, right? It will develop, it will grow, it will mature, it will get mature, right? What happened? So look, this is a daughter cell or new cell. When a daughter cell or new cell, it goes into goes for maturation. So the process of maturation or the time duration between the new cell and between the maturation, right? From the daughter cell to get a fully mature cell, fully grown cell. This time period is called interphase, right? Growth takes place during this phase. 90% of time taken by this interphase, right? From a daughter cell to get a mature cell. 90% time is taken by interphase. This is called interphase. Again, so suppose that a cell takes 12 hours to get divided. Then what will happen? 10 hours will be taken place to grow, and then 2 hours, rest of the 2 hours, that will be taken by for the division, right? And that is called M phase, right? So first what happened? A new cell undergo for the growth. It takes 90% time of the cell cycle and it grow, and that is called interphase. Further, we have divided interphase into 3 different subphases: G1, S, and G2. In G1 phase, cell organelle get double, right? In S phase, DNA synthesis takes place and protein synthesis takes place, right? And in G2 phase, protoplasm synthesis takes place, right? Then what happens when a cell get fully grown cell or become a mother cell? Now it have 3 options, right? The mature cell have 3 options. First option, it get differentiated and take a particular function, right? it get differentiated. For example, it become a cardiac cell. For example, it become a muscular cell. Whatever it want, it can become. And what will happen if it will differentiate? It will stop dividing. Then the division will stop, right? When it differentiates, then due to differentiation, the division property will stop. Then the second option, so it can opt first option. If do not want to go for differentiation, it have to go for second option. Second option is it get arrested as it is. It remains mature cell. It do not get differentiated. It remains as a reserve cell. Whenever the time will come, it will start dividing, right? So it become a reserve cell, right? The third option it will enter into G1 phase, means it will again start dividing. A mature cell will start again dividing, means it will remain in the cell cycle, right? 10% time of the cell cycle is taken for the division. Now this it enter into G1 phase, right? Division phase. So division phase, there may be two type of the division. First is mitosis, which is a very common kind of division. In mitosis, a diploid mother cell divide into two diploid daughter cell. So number of the chromosome was diploid 2n in mother cell and 2n in daughter cell. Means number of the chromosomes are after the division equal in the daughter cell as well as in mother cell. So this kind of the division where mother cell and daughter cell remains equal number of the chromosome, this is called what? This is called equational division. That's why the mitosis is also called equational division. The second one is meiosis, right? Meiosis is called, right? Meiosis is called this. Meiosis is called again the reductional division. What happened? A mother diploid mother cell, it divide into 4 daughter cell, but 4 haploid cell. So mother cell have diploid cell and daughter cell have haploid cell, right? So number of the chromosome get reduced, right? Number of the chromosome get half. So what we say, we call it reductional division, right? So in meiosis, the number of chromosome reduces, but yes, in mitosis, one mother cell gives rise to 2 daughter cell. But in mitosis, one mother cell divide into 2 daughter cell, but chromosome remains constant. But in meiosis, one mother cell divide into 4 daughter cell, and the number of chromosome get half, right? This division phase is called M phase, and growth phase is called interphase. So complete cell cycle can be divided into interphase and division phase, that is M phase, and in interphase takes 90% time, and division phase, that is M phase, takes 10%. Is it clear?
Yes, sir.
Right? Yusra, is that clear?
Yes, okay.
You know, if you want, you can take the screenshot of this one. You won't get this thing, right? This diagram just I have made to make you understand, right? So tell me. So I think it's clear to all of you, right?
Yes, okay.
Fine. So next, now again, the stages of cell cycle. So what are the stages of cell cycle? Look, this is a complete cell cycle stage. Look, a cell, look here, what happened? This is the mother cell. Mother cell divide into daughter cell. Again, daughter cell start dividing. So from here to here, this is called interphase. It takes 90% time in the interphase. Look, whenever a cell divides, so again, cell division takes place in 2 parts. The first part is called karyokinesis. Karyokinesis means division of nucleus. And the second part is that the cytokinesis means protoplasm division. Karyokinesis may be of 2 types, mitosis and meiosis. Just we have few minutes before we have understood that what is the mitosis, what is the meiosis. Again, the Fleming was a scientist who discovered mitosis, and Farmer and Moore was a scientist who given meiosis. So these are the 2 scientists. Again, look here. What happened? Whenever a cell divide, it enter into G1 phase. This is called gap 1 phase. In G1 phase or gap 1 phase, the cellular content excluding the chromosome are duplicated. So excluding the chromosome, rest of the organelle get doubled. right? Then after G1, what happened? It enter into next S phase. But before entering into next phase, there is a checkpoint. The checkpoint will what will it do? The checkpoint will cross check that is that cell is prepared enough, is that the oral organelle got doubled, and is that cell is fit enough to go in the next stage. If it is fit enough, then it will enter into S phase. Else it will be stopped at here only, right? Because defective cell formation will not take place. The checkpoint will check the quality of the these newly formed structures. So this is called CDK, cyclin dependent kinase, and this first checkpoint is called G2S checkpoint, G1S checkpoint. Sorry, this first checkpoint is called G1S checkpoint. So in G1S checkpoint, a protein called CDK, cyclin dependent kinase, it checks the quality of division, right? And then it will go ahead. So if okay, then it will go ahead. It will enter into S phase. Now, what will happen in S phase? The synthesis of DNA, synthesis of RNA, and synthesis of chromosome takes place. So basically, S phase is synthesis phase. In synthesis phase, each 46 chromosomes duplicated, they get double, and DNA replication takes place, RNA formation takes place, and the protein synthesis takes place. Right? Again, after the completion of S phase, the cell will enter into G2 phase, gap2 phase. Gap2 phase, the DNA replication stops. That is very important. It has been asked in NEET examination as well. So DNA replication stops over here, but the RNA formation means transcription and protein synthesis will keep on going on, right? Whatever happened, again, after the completion of G2 phase, now the cell have to prepare for division. It will go for division phase, right? In G2 phase, again, there is a checkpoint, G2M checkpoint. Here, G2M checkpoint, again, the protein is called CDK, cyclin dependent kinase. What it will do, it will work as a checkpoint, and what it will do, it will again function of controlling the quality of the cell. If cell is okay, then it will go ahead, and the rest, the 10% time will be given to mitosis or meiosis. This is the complete cell cycle. Is it clear? Checkpoint clear?
Yes, sir.
Jafullah?
Yes,
Clear?
sir.
Fine. Again, the same diagram have been depicted over here. Look, this is a bit more clear diagram. So here in G1 phase, like after the division, either cell will go into G0 phase, means it will stop dividing as a reserve cell or get differentiated. Else it enter into G1 phase. So growth, and again the content become double. Then interphase, there is a S phase, DNA replication, protein synthesis. G2 phase we have studied, and again here the mitotic phase means meiosis or mitosis have been divided into following phases, right? The first part is that it will enter prophase, metaphase, anaphase, and telophase. Then again it will divide, right? So checkpoint we have studied. Now here the characteristic of interphase G1, G2, G1S and G2 phase. In the interphase in G1 phase, this is the longest phase, means it takes maximum time out of the interphase. The rest of the means S and G2. RNA and protein formation will take place. Nucleotide, amino acid, histones, and ATP will be produced. Duration is variable from cell to cell, right? G1 phase duration is variable from cell to cell. Again, the second one is S phase, that is synthetic phase. In synthetic phase, what will happen? The chromosomes will replicate. DNA will become double. This is called invisible phase because this phase is not clearly visible. DNA replication takes place, but the chromosome number do not double. So they make the double content of the DNA, but they do not look double, right? So look, the like before the S phase, the chromosomes are like this one. So this is one chromosome only. Again, but there is each chromosome contain so the chromosome contain only two arms. But here what will happen when the chromosome enter into S phase, it will increase the double DNA content. The chromosome will have four arms, but it will always remains as a one unit, right? So number of the chromosome do not increases, but DNA content increases. That's why this is called invisible phase, right? And again, the kinetochore synthesis takes place, means the kinetochore is formed. Now it enter into G2 phase. In G2 phase, the synthesis of DNA stops, RNA and protein synthesis keep on going on, right? So this is called interface. Is it G1, S and G2 clear?
Yes, sir.
Check in your NCRT in case if you have a question, please ask. Then we will go ahead. Cell cycle. Then we will enter into M phase.
No doubt, sir.
Okay, fine.
Sir, there's some exceptions, right, where haploid cell go under mitosis.
Haploid cell undergo mitosis.
Mitosis. Usually, only diploid cell go under mitosis.
No, meiosis. Do you want to say meiosis? Every cell undergo due to either haploid or what happened in haploid cells. They undergo the mitosis only. Yes, haploid cell undergo the mitosis, but they do not undergo meiosis. Diploid, triploid, tetraploid, all cells go under meiosis. They undergo meiosis means the number of chromosome become half. But if haploid cell will undergo meiosis, then that is not possible, because in haploid cell, if the number of the chromosomes, suppose that there is an egg, it have 23 chromosomes. Now if it will undergo in the meiosis, what will happen? The number of the chromosome again reduce, right? Yes,
Sir.
become 11 and half and 11 and half. That is not practically possible, right? So the every cell which is haploid, that undergo mitosis, not meiosis. Meiosis is not possible, right?
Yes, sir.
Okay. So let me give you an example. Haploid organism. Have you seen honeybee? Honeybee? And we'll
Yes.
study in class 12.
Yes, sir.
Honeybee, there is a male drone. So a type of honeybee which are the male. Those male have only haploid body. They do not have double number of the chromosome. So in honeybee, there are 16 plus 16 chromosome, 32 chromosome, right? But the male have only 16 chromosome. So in male, the sperm formation do not takes place by the process of meiosis. There the sperm formation takes place by the process of mitosis. Clear, Saifullah?
Yes, sir.
That is the best example, right? So let's go ahead, right? Now mitosis. So mitosis is equational division, right? Look, this is the interphase chromosome, interphase nucleus. I'm sorry. This is the interphase nucleus. What happened in interphase nucleus? There is a structure. This is called centrosome, and centrosome. This structure, this complete structure, it is called centrosome. And the structure centrosome, it have two small T-shaped structure. These two small T-shaped structure, these structures are called centrioles, right? Look, here I have shown the diagram in detail. So here in this diagram, you can see this is the centrosome, and centrosome have two centriole which are T-shaped structure, and they are at 90 degrees of each other. This is the nucleus, right? And there is a structure called nucleolus, right? And nuclear envelope, and there is a plasma. Now it will undergo in the condensed state, right? And what will happen when it will enter in for the division? It will go into prophase. What will happen in the prophase? In prophase, these chromosome become thickened, and they are called monovalent chromosome, right? Monovalent chromosome, single chromosome, right? These both of the centrosome, like centriole, start moving toward the opposite pole, and again this nuclear envelope start degenerating. This is nuclear envelope, right? So this nuclear envelope it start degenerating. It degenerate, right? Means it disappear, degenerate. It break into fragment. After the fragmentation, what will happen? these two sister chromatids come closer to each other, right? And after that, these go into this nuclear envelope dissolve and they reaches to opposite pole in metaphase. So this is called pro-metaphase, right? Pro-metaphase, the stage before metaphase. In pro-metaphase, what happened? These centriole start formation of a spindle fiber, right? So these structure are called a spindle fiber. Spindle fiber formation takes place, and spindle fiber formation. What happened? This spindle fiber attaches with the centromere. So this is the structure. This structure is called actually chromosome. In chromosome, this middle point is called centromere, right? And this upper and lower part, this is called kinetochore. This spindle fiber attaches to the kinetochore, right? They attaches to the kinetochore, and when they attaches to kinetochore, what happens? These are pulled opposite side. They come in metaphase. They get arranged in the equatorial plane or metaphasic plate, right? When they gathered at equatorial plate or metaphasic plate, after they enter into anaphase. In anaphase, they are pulled at the opposite pole. They reaches at opposite pole. This nuclear, this spindle fiber disappear, and after in anaphase, so the division of nucleus means karyokinesis start. So karyokinesis have four phases: prophase, prophase, prometaphase, metaphase, anaphase, and last one is telophase. In telophase, these both of the nuclear, these are both of the chromosomes, they reaches at opposite pole, right? Formation of nuclear envelope takes place. Again the centrosome, like this centriole divided into two and becomes centrosome, right? Now it comes to the cytokinesis. In animal cell, what happened? Look, this is the structure of animal cell. In animal cell, what happened? The centripetal division start in protoplasm. So formation of furrow takes place from outer to inner side. Means centripetal formation of the furrow takes place, right? But in plant cell, that is something different. What happened in a plant cell? In plant cell, when the both of the nucleus divide, for a collection of a substance takes place at the center point, right? And this deposition of the material takes place between the cell, between two protoplasm, and the development of this or diffusion or deposition takes place in centrifugal plate, right, from inside to outside, right? This structure is called cell plate. So what happened in cytokinesis? In animal cell, furrow formation takes place, but in plant cell plate formation takes place. Furrow always develops centripetally, means centripetal manner, but the cell plate develop from centrifugal, from inward to outward, right? That is the only difference between the cell division of plant and animal, right? Is it clear?
Yes, sir.
Okay. The spindle fiber are made up of 4 to 20 microtubules, right? Cell plate is made up of a material called phragmoplast. Clear? So this was the
Yes, sir.
mitosis. Now check in your NCRT and let me know. Check with your NCRT. Then we'll go in meiosis. Abdullah, Saifullah, is it clear?
Yes, sir, clear.
So check in your NCRT and ask me if you have any doubt. Check and let me know when you check it. Done?
Yes, sir. So, what are the differences between in the kinesis? What's the difference between plant cell and animal cell?
That's what I was telling. Look, what happens? Suppose that this is animal cell. So in animal cell, what happens when protoplasm will divide? What will happen? This furrow formation. Look here. Look my marker. This furrow formation will take place. So from the protoplasm divide from outside to inside. Right? Clear?
Yes, sir. Yes, sir.
So that furrow will develop, and this furrow will meet at the center, and the protoplasm will divide in two parts: upper cell and down cell. Right? So this division is centripetal from outside to inside. So protoplasm division will take place in centripetal manner from outside to inside, and with the help of furrow. But what happened in plant cell? So plant cell, here look, in here at the center part, this at the center part, deposition of substance will take place, right? And this is made up of calcium pectate, and deposition of substance will take place from inside to outside. So this is the centrifugal manner, right?
Yes, sir.
This is the centrifugal manner, right? Now what will happen? This reaches to the at the margin. So when it reaches at the margin, so this structure is called cell plate, and the substance which is deposit, that is called phragmoplast, right? And this is centrifugal manner, right? So protoplasm of the animal cell divide by the formation of furrow, but protoplasm of plant cell divide by the formation of cell plate. That is the difference between cytokinesis of plant and animals. The rest of the things are same. Is
Okay,
that clear?
sir. Yes, sir. Thank you.
Okay, welcome. Saifullah, clear?
Yes, sir.
Fine. So let's go to meiosis. Look, now meiosis. In meiosis, we have already understood when I was teaching you cell cycle. So I told you that in meiosis, what happened? Meiosis, in meiosis, formation of 4 daughter cell takes place from a mother cell, right? So look, this is the meiosis first. Meiosis first is called reductional division, right? Why? Because the mother cell have diploid number of chromosome, 2n chromosome, while the daughter cell have haploid number of chromosome. So right, so number of chromosome become half. So we have already discussed that is called reductional division, because number of chromosome reduces to half. The second important thing, in mitosis, formation of 2 daughter cell takes place from one mother cell, but in meiosis, formation of 4 daughter cell takes place from one mother cell. That is also it. Again, meiosis takes place in 2 steps: meiosis 1 and meiosis 2. Now, out of these meiosis 1 is called reductional division, because here the number of chromosome reduces. Meiosis 2 is called equational division, because here the number of chromosome remains equal, right? And it is like almost equal to the mitosis process of mitosis. Look, come to the meiosis first. So meiosis first, this is prophase first. In the prophase first, these centromere divide, centrosome divide, and centriole starts moving in opposite direction. They reaches to opposite pole, right? Start moving towards opposite pole. Here, the 2 chromosomes come close to each other and make bivalent. In meiosis, the monovalent formation takes place. Only 1 chromosome are found, right? But here the chromosomes are found in pair, and this is called bivalent. So the red chromosome is different, and this blue chromosome is different. So bivalent formation takes place. Nuclear envelope disappear. So prophase, this was the prophase. In metaphase, these chromosomes get arranged at equator or at the center point, metaphasic plate, that we call metaphasic plate. In metaphase, again, these centriole reaches to the opposite pole, and they form a spindle fiber. A spindle fiber attaches with a chromosome. In anaphase, both of the chromosome are pulled at opposite pole. But here what happened, when these pair were along with each other, they have exchanged the part of chromosome. This part is called crossing over, right? We will study in detail. Again, we will study prophase in detail, right? Now from here, this was the meiosis first. Now the meiosis second. So what happened? These chromosome reaches to opposite pole. Now the number of the chromosome get half, right? These reaches to opposite pole. In opposite pole, after reaching opposite pole, the karyokinesis takes place, then cytokinesis takes place. Now one cell divided into two daughter cells, and both are the haploid cell. Now what will happen? The rest meiosis second is equal to mitosis, right? In after telophase, again prophase second come. In prophase second, again this centriole moves to the opposite pole, right? Formation of monovalent takes place, right? Nuclear envelope disappear. In metaphase, both of the chromosome remains arranged at the metaphasic plate. From metaphasic plate, in anaphase, both of the chromosomes get separate and they are pulled at opposite end. And in telophase, both reaches at opposite pole, formation of new nucleus takes place, and then formation of new cell takes place. So in meiosis, formation of four cell takes place from a mother cell, right? Formation of four daughter cell takes place from a mother cell, right? Is that clear?
Yes, sir.
Okay.
Yes, sir.
So this was meiosis, right? So now I want to share one more PPT. That is the meiosis also takes place in different phases. So prophase of meiosis is called the longest phase. So prophase of meiosis have been divided into further many points. So that's what I want to discuss.
Sir, can you please scroll up? I just wanted to take the screenshot on the previous one.
The previous slide, okay, fine. Just this one, mitosis.
Yes, sir.
Yes. Look, now you can take this.
Yes, sir.
Look, this is the diagram which I have made. These are the number of the this is one chromosome only, right? But it have the genetic material is double, right? And one more important thing I want to tell you: this center part is called centromere, and this upper part and lower part there is a disc-like protein. This is called kinetochore, right? So these spindle fiber get attached into kinetochore, not centromere. Clear?
Yes, sir.
Now come to one more point, one more PPT. Meiosis first prophase. This is the longest prophase. Again, meiosis first means prophase first, that have been divided into further 5 stages: lactotine, zygotine, pachytine, diplotine, and dikinesis. What happened? In lactotine stage, the nuclear membrane start degenerating, and the formation of these, like this chromosome, get condensed, means they become thick. In zygotine, the formation of bivalent takes place, means two chromosome comes in contact with each other, right? The part of chromosome is stuck with each other, and this stucking structure, or this structure, pairing structure, this is called synaptonymal complex. So formation of synaptonymal complex takes place, right? In pachytine, what happens? Look, this is the next stage is pachytine. In pachytine, what happened? They exchange the part, right? They exchange their chromosome part. Look, red one have given to blue one, and blue one have exchanged the part to the red one. Now, the next, so this process is called crossing over. So crossing over takes place in pachytine, means exchange of the part. So crossing over takes place. Synapsis, synaptonymal complex means synapsis takes place in zygotine. Crossing over takes place in pachytine. Now, after the crossing over, means they have exchanged the part, they start separating from each other. When they start separating from each other, they make this crisscross structure. This crisscross structure is called as synaptonymal complex, right? So synaptonymal complex, it get dissociated, and the dikinesis, that is the nuclear envelope, get dissolved, and that is the end of prophase, right? So we have further divided into lactotine, zygotine, pachytine, diplotine, and dikinesis. The number of the time the question have been asked in medical examination: when the synapsis takes place, when the crossing over takes place, when synaptonymal complex takes place. Is that clear?
Yes, sir.
If you want, you can take screenshot of this one as well. This is very clear PPT.
Done, sir.
So this was all about the this your chapter. Have a look on your NCRT. I have like we have revised the cell cycle and cell division part, right? And if it is clear, then let me know the next chapter which we are going to revise. Clear?
Yes, sir.
So, which is the next chapter we are going to revise? Tell me, all of you. Shafullah, Shahid, and Yushra, all of you. Tell me
In
what is the next.
plan growth and development, can you revise the
Yeah, sure.
plan growth, plan regulators?
Right. Definitely. We'll do that. So, let's start with the plant growth and development, plant regulator. That is the last chapter which we have studied. Plant growth. Yes, plant growth and development. Okay, fine. So let's start with the plant growth and development. Look, so first in plant growth and development, we have to study basically these three terms: dedifferentiation, differentiation, and redifferentiation. Right? Let's discuss what is the difference between all these three terms. So just right now we have studied the cell cycle and cell division. For example, this is a meristematic cell, right? A meristematic cell get mature and become differentiated. So differentiation means it will become a fully developed, fully mature cell. It will opt a particular kind of the function, means it won't divide anymore, and it will lose the property of division. It get a particular function. So this is called a differentiated cell. So first process, this is the process of differentiation. In the process of differentiation, after division, a cell get mature and get a particular function. What happen whenever the cells get damaged, right? Suppose that it become a epidermal cell, it become a heart cell, and the heart get damaged. So what will happen due to damage? The nearby cell will start again dividing. So what will happen? These permanent cell, so these meristematic cell become permanent cell. These permanent cell again regain the property of division. When again the permanent cell regain the property of division, this is called dedifferentiation, opposite to differentiation. Dedifferentiation. It again they regain the property of division. They start dividing, and cell become meristematic and divide. So this is called dedifferentiation. After dedifferentiation, so now this is the cell again regain the property of division. After regaining the property of division, it will again divide, and after division, it will again differentiate. So when it will again got a particular function and again stop dividing, this process is called dedifferentiation. So that is, there are three terms: differentiation, dedifferentiation, and dedifferentiation. Now tell me, Is it clear or not?
Yes, sir.
Safula? Right? Clear?
Yes, sir.
Yes, sir.
Now come to the plant growth curve. Look, this plant growth curve, it shows the sigmoid structure. Means sigmoid means S-shaped curve. Sigmoid means S-shaped curve. S-shaped curve. Right. So this is sigmoid curve. Now look, what is it reflect? So this in lower axis we have taken the time limit, and in the upper axis we have taken the growth of growth in the terms of size, weight of the organ and all that, right? Here what happened, in initial days a small seed germinate and a small organ develops, so the growth is slow, right? So when it develops slowly, it grows slowly. This is called lag phase. After, when it reaches to at the like young stage or juvenile state, it grow very rapidly. In short time period it start rapid growth, right? So graph will be steep, very steep. This is called exponential phase or lag phase. When it reaches to the maturity, after the maturity, now the deceleration will take place or decline will take place. This is called maturity phase. After a particular time, the time will keep on increasing, but the further growth do not take place. This is called a stationary phase, right? So sigmoid curve have been divided into four categories: lag phase, exponential phase, maturity phase, and stationary phase, right? And the time period which takes place in the growth, that is called grand time period of the growth, right? Clear?
Yes, sir.
Yes, sir.
Again, when it comes to the graph, there have been given such a particular term. So, for example, the term plasticity, right? So, what is the development? Development is nothing, just from the birth to the death of the changes between the birth to the death of a particular organism is called development. Now, what is the plasticity? This is 2 marker question for a school exam. This graph is also important. They can ask you to draw the diagram. Now, what is the plasticity? Plasticity is a property of nothing, just plasticity is a property of uh, any organism. When the change takes place in environment, it changes itself. That property is called plasticity. As the age changes, so due to the change in the age at different, like in different stages, the morphological structures are different, right? You can say when a child is like small, it do not have mustache and beard. When it grown up, it have a mustache and beard. So that is also a plasticity, change in morphological structure due to change in age, right? So these structures, this is called plasticity. So what is the plasticity? First, the change in morphological structure due to change in environment is called plasticity. Change in morphological structure due to change in the age is called plasticity. The best example of plasticity is heterophily. What is heterophily? If a plant have different type of, if the same plant have different type of the leaves in different environment and different age group, this is called heterophily. Look, this is the example of coriander, right? So in coriander, what happened in coriander? Leaf in juvenile stage have this kind of leaf. Adult is this kind of the leaf. and that those coriander which are found in terrestrial habitat, they have this kind of the leaf. Those which are found in water habitat, they have this kind of the leaf, right? So the different type of the leaf in the same plant is called heterophily, and heterophily is an example of plasticity. Is that clear or not? Please check once. Heterophily or in plasticity.
Is there any other example of this?
Just I told you, the changes in takes place in our body, in males, like a small kid when at the young age, juvenile stage, they do not have beard and mustache and hair in their body. When men develop, it become adult, it have a beard, mustache, and all that. That is also a plasticity, change in morphological structure due to change in age, right? One more thing, like those people who live in higher altitude, they have high hemoglobin level, and those lives in the plain, they have low hemoglobin level. That is also a plasticity, change in environment and the change in body structure. Clear?
Yes, sir.
Clear, Shafla and Said?
Yes, sir, clear.
Now it comes to the plant growth regulator. So plant growth regulator are basically called phytohormones. Phyto means plant and hormones and hormones. Plant hormone are phytohormones. They are also known as plant growth regulator. So the first phytohormone or plant growth regulator is called, that is auxin. The second one is gibberellin, third one is cytokine, fourth one is abscesic acid, and fifth one is ethylene. Right? So auxin, gibberellin, cytokine, abscesic acid, and ethylene. These are called phytohormones or plant growth regulator. Right? What are the function of plant growth regulator? Look, growth, differentiation, and development. Right? Growth, differentiation, and development takes place. Right? They are responsible for growth of the plant, differentiation in cells, and development of plant. The B. They differ from they differ from one organ to another organ, right? In one organ they have different impact, and another organ they have different impact. Third, what is the function of plant growth regulator? They similar response may also be seen in different plant growth regulator. Like different plant growth regulator have the similar response, right? They are required in low concentration. Site of production and site of action is different. Means they may be produced in leaf but might be functioning in fruits, right? They may be produced in flower, might be function in the leaves. So site of production and site of the function is different. These are the property or function of the plant growth regulator. Is that clear?
Yes, sir.
Yes, sir.
Now we'll study one by one. Auxin, gibberellin, cytokine, and all that. So first plant growth regulator is called auxin. Let me take one by one. So the first and foremost thing which I had told you, that they are responsible for phototropic movement. So phototropic movement we have discussed. Phototropic movement then means that is the movement in the direction of light. Movement in direction of light, like sunflower. They always have their face toward the sun. So what happened? These auxin get accumulated at opposite side of the light. Suppose that this is why the sunflower always have its face in the direction of sun. What is the reason behind it? Let me tell you. The only reason behind it that like suppose that this is sunflower. So sunflower always have its face in the direction of sun, right? So in the direction of sun, why it have? Because whenever the light falls at this region, the auxin accumulate at opposite to the light. Auxin accumulate at this region. Auxin is responsible for cell elongation. It increases the cell size. That's why this size extend spread and this side do not extend. So due to this, the sunflower moved toward the direction of sun. So in the nutshell, this auxin is responsible for movement of plant in the direction of sun. Next, it is responsible for cell elongation in shoot. It is the first discovered phytohormone, that is the plant hormone. Natural auxins are IBA, IAA. IAA is indole 3-acetic acid and IBA is indole butyric acid, right? So these are the natural auxin. It was discovered by Clark et al. The synthetic auxin, you have to remember the name of the synthetic auxin. We are using the synthetic auxin in agriculture, which has been made in the factories, and this is a chemical. The name of the synthetic auxin is 2,4-D, that is 2,4-dichlorophenolic acid. Again, 2,4,5-T, 2,4,5-trichlorophenoxy acetic acid. NAA, naphthalene acetic acid. If you have not noted down, please note it down. These are the synthetic oxygens. It's very important. They have been asked in medical exam. Now I will tell you the function of auxin. It stimulates respiration, responsible for cell enlargement, means increases the size of cell. Responsible for xylem differentiation, means formation of xylem takes place. Responsible for cell division in cambium, right? Intrafascicular, interfascicular cambium. Responsible for cell division. Responsible for root initiation. Yes, it is responsible for root initiation. So in a stem cutting, before grafting it, what we or before rather than before implanting it, what we do, we just dip it and put it somewhere else, right? It is responsible for apical dominance. Apical dominance means like the growth will take place in the apex part, but the lateral growth do not take place. It is responsible for phototropic movement, means movement in the direction of light. It is responsible for formation of female flower in plant, right? So if we spray auxin, it will make more female flower in comparison to male flower. Is that clear?
Sir, can you please explain that last point once more?
A female flower. So suppose that we have a hermaphrodite plant, or rather than if we have a plant which have both male and female flower. So if we spray auxin, means it will produce more female flower in comparison to male flower. So number of female flower will be more. So what will happen? Means productivity will be more because the fruit formation will take place from female flower, right?
Yes, sir. Okay.
Clear? Shafullah and Said?
Yes, sir.
Great. Now next is Gibberellin. This is the Gibberellin. Gibberellin is weakly acidic hormone. It increases internodal growth. Means these are the node. Every stem have these kind of the structure. These are called node, and the distance between the two nodes is called as internode. Right? It increases the internodal growth. It was discovered by Kurosawa and Hori in a fungi called Gibberella fusariaceae. Gibberella fusariaceae is a fungus which causes a Bakane disease in Japan. Bakane disease is also known as foolish seedling disease. Means the formation of seedling takes place, but formation of seed do not take place. Right? So it was discovered in Bakane disease takes place in rice. So it was discovered in the Bakane disease of rice. Right? That is also called foolish seedling disease. And what is the function of Gibberellin? Basic function we have studied, it increases the internodal growth. Growth in stem, leaves, and aerial part. So it increases the growth of the aerial part. It is responsible for bolting. What is the bolting? Bolting is the increasing the length of a genetically dwarf plant. Example, for example, this is a cabbage, right? So cabbage have very small internode, and it have number of the leaves at the same point. That's why it's a bunchy structure. It's a rosette structure. Whenever in a cabbage we spray this gibberellin, what happen? The length of cabbage is hardly, it is hardly 9 inch in length. So whenever you spray the gibberellin, the plant become 5 feet plant. This is the diagram of that 5 feet plant. That is a cabbage. It's a cabbage plant. So the process of increasing the internodal growth is called, that is vaulting. This practice is called vaulting. This is responsible for overcome the natural dormancy of buds, tubers, and seed. It is responsible for flowering in long day plant, right? It promote the male flower. So we have studied that Auxin spray, it promote the number of female plant, but it promote the formation of male flower, right?
So, sir, does it increase the size of the leaves as well?
Sorry, increase in?
Does it increase the size of leaves as well?
Yes. It is responsible for growth of stem, leaves, and all aerial part. It increases the size. So suppose that if you are growing spinach, so what you are growing, spray this hormone, what will happen? The coriander leaves will be larger. The spinach leaves will be larger. That's what it do. And that's how the actually phytohormones are used in agriculture. Tell me, guys, is that clear? All of you?
Yes, sir. So, sir, does it have any side effects or any harm?
Yes, it do have. Nowadays in agriculture, number of the phytohormones are used. They are like, look, there are two kind of the phytohormone. Those phytohormone which are expensive one, they biologically degrade, right? They are biodegradable. Means whenever you spray, they drop their impact, and after some time, after 15 days, 1 month, they degrade automatically. So they are not harmful. But what happened? But they are very costly. So there are some cheap substitutes. They drop the impact on the vegetable and fruits, and after the dropping their impact, they remain in the vegetable as such. Whenever a person cook and eat, then they are very harmful, right? So there are phytohormone. They are beneficial as well as, but they are harmful as well as, right? So next one is cytokine. Cytokine is basically responsible for cell division, right? So the cytokinin is basic in nature, right? It was discovered by scientists known as Skoog and co-worker, and they promote the cell division, right? They are responsible for promoting the cell division. Their function is cell division, cell elongation, and auxin plus cytokinin, they are responsible for or formation of new organ. So that is called morphogenesis. So what happened during the tissue culture, when we want to like in tissue culture, we take our tissue from any plant, and what do we do after that in lab? We start putting some hormone, and they start dividing with the help of mitosis. After some time, a mass of undifferentiated cell takes place. Now what we do, we now we want in that part, that mass is called callus. That undifferentiated mass is called callus. Callus is undifferentiated mass. Now what happened when we put the auxin and cytokinin? The formation of new organ takes place. That is, the formation of leaves takes place, formation of root takes place, and the process is called morphogenesis, right? Next, they are responsible for delaying senescence, right? They delay the senescence, means they delay the death, right? They are responsible for overcome the seed dormancy. They are responsible for the overcoming the seed dormancy. Means the seed they remains inactive, and whenever we put the auxin, the seed come out of the activation part, the activation mode, and they come out of dormancy and they germinate. They provide resistance to temperature, right? Yes, they are responsible for providing resistance to temperature to the plant. Plant with the more auxin, they can more cytokinin, they can withstand in high temperature. they induces the parthenocarpy, means that is the formation of that is seedless plant, seedless fruit, right? So that is the function of cytokine. Have a look and let me know. Is that clear?
Yes,
Sir,
sir.
so they work against Auxin, sir.
No, they work with Auxin actually. They work with Auxin. You know, actually, cytokinin do not work alone. It requires the help of Auxin. That's why I have written over. Look, when we mix the Auxin and cytokinin, then what they do, they are responsible for morphogenesis. Yes, they work along with Auxin. Clear? Shahid? Shahid, is it clear?
Yes, sir.
Okay, great. Safullah, clear? So let's move in next hormone. Next is ethylene. Hello. Ethylene stimulate the transverse growth, means it is responsible for the growth of the branches, right? Maximum synthesis occurs during the ripening of fruit because this is responsible for ripening of fruit, right? Function, ripening of fruit. And one more thing, this is the only gaseous hormone. This is the only gaseous hormone, right? The function, ripening of fruit. It hastens the senescence of flower, means if you will spray the ethylene, then the flower will get down, right? They will detach, right? They will die. It breaks the dormancy of seed and bud. Yes, they are also responsible for breaking dormancy. Uh, they are responsible for uh, uh, elongation of rise. So they increases the length of right, and they are responsible for root initiation. So they are they are used in stem cutting, and they stimulate the flowering of pineapple and mango. So this question I have been asked in medical examination 2 or 3 times I have seen. So because like which hormone is responsible for stimulating the flowering in pineapple? So this hormone is responsible for stimulating the flowering in pineapple plant, right? Is that clear?
Yes, sir.
Yes, sir.
Next is abscesic acid. In short, we call it ABA. Look, abscesic acid. ABA is also known as stress hormone, right? It is also known as dormant, right? Because it is responsible for dormancy. Abscission, it is responsible for abscission of flower and fruit, right? Look, abscisic acid. Right? Abscisic acid is known as a stress hormone. Very important. Why we call it stress hormone? Because it protects plant during the stress situation. What kind of scarcity of water, high temperature, high water content, and all that, right? That's why. So whenever the plant comes in stress situation, right, excess of rain, the temperature become high, the drought, water got stress. So that's why this is called a stress hormone. It is also like, for the sake of example, this is the water scarcity. So more abscisic acid formation will take place. Now what will happen? It will reduce the rate of transpiration, right? Rate of transpiration reduces. So due to decrease in the rate of transpiration, it will save water and make the plant alive for longer time. Next one, it is also called dormant because it is responsible for dormancy of seed. The third one, it is responsible for abscission of flower and fruit, right? Next, it reduces the transpiration. The rate of transpiration reduces, and it is responsible for flowering as a strawberry, right? So that's why, that's what the function of abscisic acid. Finally, it is responsible for defoliation of leaf, fruits. So when the leaf get mature, they fell down. That is called defoliation. When the fruit get mature, they fell down. How does they fell down? Look, whenever leaf or fruit get mature, the formation of abscission layer formation takes place. What is the abscission layer? Abscission layer, that is the death of tissue takes place. So dead layer is formed between the stem and petiole, or stem and petiole of the fruit. So dead layer of dead tissue is formed. That is called abscission layer. Abscission layer formation takes place due to abscisic acid, right? And this abscisic acid is responsible for defoliation of leaves and defoliation of this ripened fruit, right? Tell me, is it clear?
Yes, sir.
What is the photoperiodism? Look, photo means light and period means duration. So effect of duration of light on plant is called photoperiodism, right? So term is photo plus period and plus is. So photo means light, right? And period means duration, and is, that is impact, right? So this is called the photoperiodism. So whenever we study the impact of duration of light time period on plant is called photoperiodism. It was discovered by Garner and Allard in a tobacco plant, and the name of that tobacco variety was Monovalent Mammoth. That question I've been asked in medical exam. So Garner and Allard discovered the impact of light duration on the plant in tobacco plant, and the variety of that tobacco plant was Monovalent Mammoth. On the basis of duration of the light, the plant have been divided in basic three categories: short day plant, long day plant, and day neutral. Short day plant, in short, they are called SDP. They require a small photoperiod. That is, basically, they flower in the winter time period when the days are smaller. Days cannot be smaller. Day is 12 hour and night is 12 hour. What is the smaller? They basically, the SDP means short day plant, means they are responsible for the flowering and in the winter, means short days, when the days or photoperiod is short. The example is xanthium, chrysanthemum, dahlia, rice, sugarcane, strawberry, potato, and tobacco. All these plants are grown in winter time, right? And they flower in winter time. Look, You will frequently see these questions in medical examination. They will give you example like xanthium belongs to short day plant, long day plant, dough neutral. So anyhow you have to remember these name of the example of SDP, LDP, and day neutral. Next one is long day plant. Long day plant, they are henbane, wheat, oats, sugar beet, radish, barley, etc. Right? So long day plant are those which basically flower in the summer time period. Means they require long photo period, approximately 9 to 10 hour light. So it depends on plant to plant, but I'm just telling you an average idea, 9 to 10 hour light, right? So long day plant are those plants which require a long time for the light period. Example, henbane, wheat, oats, sugar beet, radish, and barley. Then now next one, it comes to the day neutral. So day neutral plant are those plants which do not have any impact in of the photo period. They flower throughout the year. They produces their product throughout the year in all 12 months. Example, tomato, pepper, cucumber, pea, sunflower, maize, cotton, etc. Tell me guys, is that clear? Yushra, Shafula, Shahid, is it clear? SDP, LDP, and day neutral plant.
Yes, sir.
Take the screenshot and keep it in your mind. That example, you must know the example because to get the NEET examination, right? To capture that question, you have to remember that. Last but not the least, last topic is vernalization. What is the vernalization? Look, the scientists have seen that many of the plant they require low temperature treatment. Whenever we give low temperature treatment, so whenever they get the low temperature treatment, then what happens? They flower and they produces the their productivity, like they flower. So to get early flowering in them, to reduce their lifespan, right? So before sowing the seed, we give it low temperature treatment. So giving the low temperature treatment to any plant before the sowing is called vernalization. And vernalization, we give the temperature 0 to 5 degrees centigrade to every seed and before sowing. And whenever suppose that a plant have a lifespan, like a plant takes 16 a week in flowering. So that is the basically. So whenever we use the vernalization process, then the flowering will take place only in 6 weeks, right? That's how we reduces the reproductive time period, the reproductive life of the plant. This process is called vernalization. Tell me, guys, we have completed the we have revised the complete chapter. Tell me, is it clear or not? Shafullah, is it clear?
Yes, sir, it's clear.
Okay. Tell me the next topic which you want.
Sir, photosynthesis.
Photosynthesis.
Yes, sir.
Okay. Fine. Look, light reaction means photolysis of water. So here we are studying photosynthetic pigment system, right? PS system. You can see the pigment system are of two type, PS1 and PS2. These are the structure of grana, right? These are the thylakoids, right? This one, this complete structure is called grana, right? And this indivisible, this is called thylakoid. They are called thylakoid, right? And the region of thylakoid which are stuck with one another, these part are called operous part. These are called operous part. And the outer side part, external part, which are not stuck with each other, that is called non-operous part. The photosystem first are found in non-operous part, and photosystem second are found in operous part. First important point. The second important point, the breaking of water molecule takes place means in operous part, where in photosystem second. The water molecule breaks down and formation of hydrogen ion and oxygen molecule takes place, right? In photosystem second, they have again one more important thing, that is called oxygen evolving complex. So oxygen evolving complex help in the photolysis of water, that is the breakage of water, right? So this is the photosystem structure. This is the structure of photosystem, right? Clear? This concept get clear to you? Got it?
Yes, sir.
Yes, sir.
Now come to the photosystem, the structure of photosystem, right? We have studied the photosystem 1 and photosystem 2, and now we are studying here what is the photosystem. Just common photosystem. What is the photosystem? Photosystem is basically made up of three things. First, the reaction center. That is, reaction center is made up of a special type of chlorophyll A molecule, right? And then the primary electron acceptor, right? So these chlorophyll A molecule and primary electron acceptor both combine and makes a structure that is called photo reaction center, right? Around the reaction center, there are some small pigment. These are pigment molecule. They are called antenna molecule. Antenna molecule, they capture small amount of the energy and they transfer finally energy to reaction center. Reaction center breaks down the water molecule and gives electron to electron transfer system, right? PS1 is found in non-operous part, outer side. They have more chlorophyll A molecule than chlorophyll B and carotenoid. So number of chlorophyll A molecule is more in photosystem 1. They have photocenter and electron carrier. Photocenter is made up of a dimer of chlorophyll A molecule, means 2 chlorophyll A molecule, right? So this is made up of dimer of chlorophyll A molecule. That is called, the name is that P700. Chlorophyll B and carotenoids are also present. It takes part in both cyclic and non-cyclic photophosphorylation. Tell me, is that clear, PS1?
Yes, sir.
The next one is PS2. Photosystem second, this is found in operous part. They have chlorophyll A, chlorophyll B, and carotenoids, all in equal amount. In PS1, the chlorophyll A molecule was more. Here, all found in equal amount. They have carotenoid higher than PS2, PS1, right? So they have more carotenoid pigment. They have photocenter, Auxin evolving complex, and light harvesting complex, LHC, right? Auxin evolving complex, it breaks down water molecule with the help of magnesium ion, and that is called photolysis of water. Photocenter is made up of a special kind of chlorophyll A molecule, that is dimer of chlorophyll A molecule, that is P6H. Auxin evolving complex is made up of manganese, calcium, and chloride. Is that clear?
Yes, sir.
Electron transport chain. Electron transport chain takes place in here in chlorophyll, right? In thylakoid membrane. This is the structure of chloroplast: outer structure, outer membrane, inner membrane, and inside membrane there are some this pile-up structure. These are called grana. Grana are made up of small coin-like structure called thylakoid. Thylakoid have a special kind of structure in their membrane that is called PS second and PS one. Right. Next is electron transport chain. Look, this is called non-cyclic electron transport chain. It is more important. Non-cyclic. What happened? Light energy falls down in these antenna molecule. P700, P680 is reaction center, the photosystem second. Photolysis takes place. Electron get ejected. They reaches to P680. From P680, electron reaches to pheophytin, then plastoquinine, then cytochrome B6F complex. Whenever the electron passes through cytochrome B6F complex, formation of ATP takes place. Then it reaches to plastocyanin, then P700. P700 gives again energy to electron. Electron jump. They reaches to chlorophyll A molecule, right? Then phylloquinine, then iron sulfur complex, then ferridoxin, and finally NADPH receives the electron. So NADPH is final acceptor of electron. This NADPH will be utilized in Calvin cycle. Again, this ATP will be utilized in Calvin cycle. So this is called Z scheme because this structure is called Z-like structure. It looks like Z-like structure, right? This is called Z scheme, and this is called non-cyclic photophosphorylation because electron get originated from one point and it is consumed at another point. So this is called non-cyclic photophosphorylation. Is that clear?
Yes, sir.
Yes, sir.
Now come to the cyclic photophosphorylation. Right? In cyclic photophosphorylation, when NADPH is not there to receive the electron, what will happen? Electron will come back. Electron will originate from P700, right? It reaches to the primary acceptor. Then it comes to the ferridoxin. After ferridoxin, it should go to the NADPH, but NADPH is not present. So it will again comes back to plastoquinine, then cytochrome. So when it from ferridoxin it comes to plastoquinine, generation of ATP takes place, right? Again, from plastoquinine comes to cytochrome complex. When it reaches to cytochrome complex, again ATP formation takes place. Then plastocyanin, and again then come to P700. So electron originate from P700 and again comes back to P700. So this is called cyclic photophosphorylation, right? Because electron is coming cyclical, right? From where the electron has started journey again came back to the same point, right? Clear, guys?
Yes, sir. Clear.
Yes, sir. Clear.
So keep it till here. We have already covered 2.5 chapter, right? And give the exam of 2.5 chapter. So I want to ask you, like, when you have your exam, Yushra?
So bio exam is tomorrow.
Bio is on 12.
Tomorrow.
Tomorrow. Okay.
Yes, sir.
So you can ask me if you have any extra doubt
Yes, sir.
in case. What about you, Shafullah? Bio exam?
Biology pre-board is on Wednesday. This
Middle of
Wednesday.
or Wednesday? Even you should ask. What about you, Shahid?
6th, sir.
6. Ask me if you have those are two people who have their exam. Ask me if you have any doubt.
Sir, can you please explain me Kelvin cycle?
Yes, Kelvin cycle. Why not? Look, this is called Kelvin cycle. Kelvin cycle is just, it takes place in the matrix of chloroplast. Look, if when one time Kelvin cycle moves, it fixes one carbon molecule, and glucose molecule have 6 carbon molecule. So to make a glucose molecule means it have to fix 6 carbon molecule. So how many times Kelvin cycle will operate? 6 times, right?
Yes, sir.
Okay. There is a enzyme, there is a substance called RUBP. It is 5 carbon compound. It react with carbon dioxide. So primary acceptor of carbon dioxide is RUBP. RUBP combined with, so 5 carbon plus 1 carbon, total 6 carbon. RUBP combined with Rubisco, RUBP combined with carbon dioxide and makes the that is the 2 molecule of 3 carbon compound, total 6 carbon compound. That is called PGA, phosphoglycerate. To 2 molecule of phosphoglycerate are formed. Phosphoglycerate changes into G3P, glyceraldehyde 3-phosphate. During this process, consumption of 2 ATP will take place. 2 ATP is spent, 2 NADPH is spent, right? And this process is called reduction, right? Here reduction takes place, right? Then formation of 2 molecule of G3P, glyceraldehyde 3-phosphate takes place. Out of, so there are, it is again 3 carbon compound, 2 molecule of 3 carbon compound, total 6 carbon atoms are there. Now what happened? 1 carbon get detached over here, it goes to the glucose. So it gives 1 carbon to glucose formation. What will happen? Now the 5 carbon are left, right? So formation of ribulose biphosphate takes place, RP, right? Ribulose phosphate takes place, that is called RP. Now this is the 5 carbon compound because it have given 1 carbon to this one, glucose. And again, regeneration process is again the formation of RUBP. Again, ATP changes into ADP. Again, 1 phosphate it takes a place and become like RP takes place, 1 phosphate and become RUBP, ribulose biphosphate. So again, the formation of RUBP takes place. That's why this is called regeneration. So Kelvin cycle takes place in 3 processes: carboxylation, that is addition of carbon dioxide; reduction, formation of NADP, right? And again regeneration, again the formation of RUBP. This is called Kelvin cycle. Is it clear, Krishna?
Yes, sir. Thank you, sir.
Welcome, madam. Shaifullah, is that you have any doubt?
No, sir.
No? Fine. So, Shahid, you have on 6th of the this month, right? So, today is 28th. So, you have 5 days. In case if you have any doubt, any problem, so you can talk to Nisra ji and you can discuss with me and you can take the class, right?
Yes, sir.
Fine. So, all clear?
Yes, sir.
Shaifullah, all clear?
Yes, sir.
Fine. So keep it till here, right? And let's meet someday next class, and we'll discuss the new topics. Clear?
Yes, sir.
Fine. So bye bye. Good night. All the best to all of you, right, for your exam.
Thank you,
And
sir.
I hope may you all come with the flying color with the good marks, right? Fine. Shahid, Shafullah, and
Yes,
Isha.
sir.
Bye bye. Take care.
Thank you, sir.